Hello, my name is Joshua Mugabi. I welcome you to Biology Remedial Lessons. Today we are on Remedial Lesson 8 and we are going to discuss osmosis. You are welcome. What is osmosis? What is osmosis? We can say osmosis is the movement of water or solvent molecules from a dilute or hypertonic solution, in our diagram, that is part A, to a more concentrated or hypertonic solution, that is part B, across a semi-permeable membrane. So when you look on the diagram, you will see that in the middle we have a partially permeable membrane. We have the water molecules and we also have the solute molecules. On side A, we have more water molecules than on side B. And on side A, we have less solute molecules than side B. That means that side A is more concentrated. More concentrated with water molecules, and side B is less concentrated with water molecules. But if we are looking at the concentration, we always consider the solute concentration. So that means that side A is less concentrated. That means that it is dilute or hypertonic. And side B is more concentrated with solute molecules, and that means that it is hypertonic or more concentrated. So our osmosis would mean that water is going to move from the dilute hypertonic solution, that is A, to a more concentrated hypertonic solution to side B across a semi-permeable membrane. Still, on osmosis, we can say is the movement of solvent molecules from a region of their high concentration to a region of their low concentration through a semi-permeable membrane. We can say osmosis is the movement of solvent molecules from a region of their high concentration to a region of their low concentration through a semi-permeable membrane. Let's look at our diagram. We have side A and side B. Side A has a high concentration of solvent molecules, which is water. And side B has low concentration of water molecules. Therefore, water, which is our solvent, is going to move from side or region A to region B through a semi-permeable membrane. So we can say that osmosis is the movement of the solvent molecules from a region of their high concentration, that is side A, to a region of their lower concentration, that's side B, through a semi-permeable membrane. State the factors affecting osmosis. State the factors affecting osmosis. The first factor is the concentration of the solution. The concentration of the solution. Let's see it on our diagram. We have side A and side B. Side A, we have more water molecules. And on side B, we have less water molecules. But on side A, there are no sugar molecules. And on side B, we have more sugar molecules. That means, in terms of concentration, if we are looking at the sugar molecules, side B has more sugar molecules, therefore it is concentrated compared to side A. But when we talk of the water molecules, we have a high concentration of water molecules on side A than on side B. Because on side B, the sugar molecules are attracting most of the water molecules, and that one reduces the concentration of water molecules on side B. So that means that on side A, we have high water molecules, high concentration of water molecules, and on side B, we have less concentration of water molecules. And because of that, water will move, or osmosis is going to take place from region A going to region B through a semi-permeable membrane. So the concentration is one of the factors that will affect osmosis. We are still looking at the factors affecting osmosis. Another factor is the concentration gradient. Still looking at both sides, side A with more water molecules, and side B also has water molecules. But the difference between side A and side B is that on side B, we have solute molecules or solute that cannot go through the semi-permeable membrane. And those solutes attract the water molecules around them. And when these water molecules are attracted to the solute, it means that now they are no longer free to move. So the amount of water molecules that are free to move reduces. And this one also reduces their concentration. So now we can see that side A has got a higher concentration of water molecules that are free to move compared to side B. So with that one, now we have created what we call a concentration gradient. So water will move from region A, where there is high water molecule concentration, to region B, where there is low water molecule concentration. So that concentration gradient is one of the factors that will affect osmosis. Another factor affecting osmosis is temperature. If we look at our container, side A and side B, osmosis has been taking place. And water molecules have been moving from region A to region B. Osmosis continues. It only stops when an equilibrium is reached. Now let us suppose that an equilibrium has been reached. And we apply heat on side A. If we apply temperature on side A, that temperature is going to provide kinetic energy to the water molecules. And these water molecules will now move faster. So they will be able to pass through the semi-permeable membrane and go to side B because of the high energy provided by temperature. So another factor which is affecting osmosis is temperature. The higher the temperature, we expect osmosis also the rate to increase. Explain the role of osmosis in a living organism. Explain the roles of osmosis in living organisms. 1. Osmosis helps to draw water into roots of plants. Osmosis helps to draw water into roots of plant. Now let us look at our root hair. It is a thin permeable cellulose cell wall, and it has a partially permeable cell surface membrane, and of course inside the hair cell there is more concentrated solution in the cytoplasm and the vacuole, meaning that there is lower water potential. By lower water potential, we means it is hypertonic in the cytoplasm of the root hair. Now outside the soil, we have a high water potential, meaning that it is dilute. There is more water outside. So water is going to move from the soil into the root hair by osmosis. It will diffuse down the water potential gradient. So osmosis assists the movement of water from the soil into the root of plants. Another one, osmosis helps in the passage of water from one living cell to another in the plant. It helps in the passage of water from one living cell to another in the plant. Now, if you look at our diagram, we can see we have our root hairs connecting to other cells which are connected to the xylem vessel. So when water enters into the root hair, it will move through the cell of the root hair and it will go into another cell next to the root hair. And then that one will give the water also to another cell which is next to the xylem, until when water enters into the xylem vessel and it is transported. Now, water is moving from one cell to another by osmosis. And this one means that osmosis is helping in the passage of water from one cell to another. Another role of osmosis in living organism is that it helps to keep the plant cell tagged, therefore increasing support. It keeps the plant cell tagged, increasing support. So when we have our cell, the cell sap is hypertonic and the surrounding area is hypotonic. So water will move by osmosis from the surrounding area and enters into the cell vacuole. And when that one happens, the cell vacuole is going to expand and it will push the cytoplasm towards the cell wall. And that one is going to create a pressure inside, which we call the tag pressure. And we say that the cell is tagged. And when the cell is tagged, that means that now it can provide support to the plant because the cell is tagged. But what brings about taggedity is the movement of water into the cell by osmosis. So osmosis helps to keep the plant cell tagged and thus increasing support. Another one is that osmosis helps in the opening and closing of the stomata. It helps in the opening and closing of the stomata. Now, this is how it happens. High concentration of potassium inside the guard cell is going to lower the water potential in the guard cell. And because of this, water is going to move in the guard cell by osmosis down the water potential gradient. The entry of water, this one increases the volume of the guard cells. So they expand. The thinner outer wall expands most. So the cells curve apart. And when that one happens, the stoma opens. And our guard cell now, all the stomata is open. What is bringing about the opening of the stomata is the movement of water from outside into the guard cell, and it is moving by osmosis. So osmosis helps in the movement of water and it helps in the opening and closing of the stomata. Another role of osmosis in living organism is found in feeding in insectivorous plants. Insectivorous plants, these are plants that are growing in areas with low nutrients or minerals. So they trap insects for their source of minerals. And this one is due to osmosis. When the insect lands onto the leaves of the insectivorous like Venus fly plant, the leaves close very fast and close the insect inside the leaf. And then they produce enzymes which digests the insects, and the insect provide the mineral nutrients to the plant, which are absorbed through the leaves. But for them to be able to trap the insect inside is by osmosis. Movement of water very fast inside and outside of the cells causing the leaves to fold very fast, and it holds the insect inside. So osmosis is used in feeding in insectivorous plants. A group of students set up an experiment to investigate a certain physiological process. A group of students set up an experiment to investigate a certain physiological process. And below is our setup. There is a beaker with water, and there is a raw peeled potato which has been cut into a curve on top, and they have put there sugar crystals. After some time, they observed that the level of sugar had risen. After some time, the students observed that the level of the sugar had risen. What was the physiological process under investigation? What was the physiological process under investigation? The physiological process under investigation was osmosis. It was osmosis. Was there a rise in the level of sugar solution? Why was there a rise in the level of sugar solution? The sugar solution is more concentrated than the cell sap. The sugar solution is more concentrated than the cell. Those cells that are next to the sugar solution become more concentrated and therefore draw water from the neighboring cells. Those cells next to the sugar solution become more concentrated, and therefore they will draw water from the nearby cells. This process continues until the cells in contact with water in the container draw it up, and this causes a rise in the level of the sugar solution. So the process will continue until the cells that are in contact with water in the container will draw it up, causing a rise in the level of the sugar solution. Suggest the results that the students would obtain if they repeated the experiment using cooked potato. Which results would the student obtain if they repeated the experiment using cooked potato? The level of the sugar solution will not rise. The level of the sugar solution will not rise. That is, if the students repeated the experiment using cooked potato. Because boiling kills or destroys cells, making them osmotically inactive. When we cooked the potato, it killed and destroyed the cells and made them osmotically inactive. Therefore, there is no water movement towards the sugar. Therefore, the level of the sugar solution cannot rise. Explain the following terms. Explain the following terms. Hypertonic. Hypertonic, a solution whose concentration is lower than that of the cell. A solution whose concentration is lower than that of the cell. For example, if we look at the plant cell, if you put it in pure water or solution with a high water potential than the cell, therefore, the pure water is referred to as hypertonic, and the water will end up entering into the plant cell. The same applies to the red blood cells. If it is dropped into pure water or dilute solution, that is a solution with low concentration of solute molecules, water is going to be drawn into the red blood cell because the surrounding water, which is surrounding the red blood cell, is hypertonic. And this will cause more water entering into the red blood cell, which is going to cause it to burst. Isotonic. Isotonic. This is a solution whose concentration is the same as that of the cell. A solution whose concentration is the same as that of the cell. In plant cells, the solution in the water has the same water potential as the solution in the cell. The same applies to the red blood cells. The solution in the water is the same concentration as the solution in the red blood cell. So all the cells will remain the same because they have got the same concentration, which we call isotonic. Hypertonic. Hypertonic. A hypertonic solution is a solution whose concentration is higher than that of the cell. Is a solution whose concentration is higher than that one of the cell. Let's look at the plant cell. In the plant cell, the solution in the water or in the container is hypertonic, meaning that it is highly concentrated compared to the solution in the cell. And because of this, it is going to cause the water to move from the cell into the water in the container. And this is going to cause a plasmolyzed cell. In the animal cell, water will move from the cell into the solution in the container because the solution in the container is concentrated. It has high concentration of solute molecules compared to the solution in the red blood cell. And because more water is moving away from the red blood cell, it is going to cause the red blood cell to shrink. Therefore, we say that the solution in the container is hypertonic because the solution has got a higher concentration than the solution in the cell. tag pressure. Tag pressure. As a cell gains water, if we look at our cell there, the cell is gaining water, that is movement 2. Its virtue in number 1, it enlarges and it exerts an outward pressure, that is pressure 3, which is called the tag pressure. So when the cell takes in water by osmosis, that is number 2, the virtue enlarges. The virtue is number 1. It goes on enlarging as the cell takes in water. Now, the virtue enlarging, it exerts an outward pressure onto the cell wall. That outward pressure, which is exerted by the virtue on the cell wall, is what we call the tag pressure. That is pressure 3. Plasmolysis. Plasmolysis. If a plant cell is placed in a hypertonic solution, that is solution 1, it loses water. The protoplasm, that is number 2, shrinks to an extent that it pulls away from the cellulose cell wall, number 3. That's what we call plasmolysis. You have a plant cell, you place it in a hypertonic solution, a solution which is concentrated. The plant cell is going to lose water. And as it loses water, the protoplasm, that is the cytoplasm and its content, shrinks. It becomes smaller. That is, it pulls away from the cellulose wall. And therefore we say the cell has been plasmolyzed. Plasmolysis. Wilting. Wilting. When a plant is tagged, it can stand upright. However, if the cells lose a lot of water, taggidity is reduced. The plant then, after losing its taggidity because the cells have lost a lot of water, the plant droops because the cells are now flaccid. Therefore, we say the plant is said to wilt. So wilting, the plant will droop after losing a lot of water from its cell. So the cells become flaccid. And when the cells become flaccid, the plant is going to droop. And then we shall say the plant is wilting. Hemolysis. Hemolysis. If red blood cells are placed in distilled water, the cells take up water by osmosis. They swell and they end up bursting. That process is what we call hemolysis. Red blood cells are placed in distilled water, meaning that distilled water is pure water or dilute solution with low concentration of solute compared to the red blood cell. The red blood cell will take in water by osmosis. And as they do so, they will swell and they will end up bursting. That is what we call hemolysis. This is because the red blood cells do not have any mechanism like the cellulose cell wall to prevent overstretching, nor any means of removing excess water. So that one means that the red blood cells are going to end up, and when they burst, because they don't have a cell wall to prevent them from bursting or any other mechanism of removing water, the only thing which is going to happen is going to be the bursting. This is called hemolysis. A Form 1 student placed red blood cells in different salt concentration and obtained the following results. A student placed red blood cells in different salt concentrations and obtained the following results. In the first one, there was a positive gain in the volume. In the second, there was no change, meaning there was zero gain in volume. In the third, there was loss in volume. So explain these results. Now, in the first solution, where there was a positive gain in volume, the red blood cell absorbed water by osmosis. This caused them to swell and they ended up bursting, which we call hemolysis. Hence, that means that the solution was hypotonic. The solution was hypotonic. In the second solution, there was no change in size or structure, which means that the solution was isotonic. The solution had the same concentration as the red blood cell. So there was no osmotic gradient, and therefore there was no change in volume. In the third solution, the red blood cell lost water to shrink, hence became crenated, as the solution was hypertonic to the cell cytoplasm. This means that in the third solution, the red blood cells lost a lot of water because the solution was hypertonic, and therefore they became crenated. What factors affect the rates of osmosis in living organisms? What factors affect the rates of osmosis in a living organism? The first one is temperature. Increase in temperature is going to increase the kinetic energy of water molecules, making them to spread faster. Increasing temperature increase the rate of osmosis and vice versa. Another factor is the concentration gradient. It is the difference in the concentration on either side of the semi-permeable membrane. That is what we are calling the concentration gradient. The difference in concentration on either side of a semi-permeable membrane. The higher the osmotic pressure difference, the higher the rate of osmosis. So osmosis is going to take place if one side has got a higher osmotic pressure compared to the other side. Explain briefly the role of osmosis in living tissues. Explain briefly the role of osmosis in living tissues. We shall start in plants. Osmosis facilitates the absorption of water from the soil by plant roots. Water is required for the process of photosynthesis. So when you look at our diagram, we have the root hairs. And the root hairs are surrounded by soil particles with film of water around the soil particle. The root hairs will absorb the water from the soil by means of osmosis. And this water will move through the root hair to the next cell by means of osmosis, until when it moves to the xylem, where it will be transported up the plant. So osmosis facilitates the absorption of water from the soil by plant roots, and this water is carried to different parts of the plant where it is required for the process of photosynthesis. Still in plants, osmosis leads to the turgidity of the cells, which contributes to the support in herbaceous plants, and it also helps the plant to maintain their shapes. If you look at our diagram, the cell takes in water by osmosis, that is number 2, and the virtue will increase in size. As it increases in size, it provides a pressure, number 3, on the cell wall. That pressure we call the tag pressure. And this tag pressure exerted on the cell wall makes the cell to become tagged. So osmosis contributes to the turgidity of cells, which contributes to the support in herbaceous plants, and it helps the plant to maintain shape. Osmosis also helps in the closing and opening of the stomata, thus regulating the process of gaseous exchange and transpiration. Of course, the stomata will close when there is little water in the stomata. But as the potassium ions enter into the stomata, That means that there is low water potential. And when this one happens, this will attract the water to enter into the stomata due to the presence of potassium ions. And when water enters into the stomata, it is going to cause the gut cells to expand. The outer layer expanding more than the inner layer. And this one will cause the stomach to open. And the stomata will open. All this is because of the movement of water from the neighboring cells into the gut cell by osmosis. So osmosis helps in closing and opening of the stomata. And this one will regulate the process of gaseous exchange and transpiration in plant. Osmosis also facilitates feeding in insectivorous plants like Venus flytrap. These are plants that depend on insects for nutrients like minerals. When the insect lands onto the leaves of the Venus fly, by osmosis, water is withdrawn and taken into the cells, and then the leaf will close very fast. And the insect will be trapped into the leaves. And then the leaves will secrete enzymes which will digest the insect, and then they will absorb the digested nutrients into the cells of the leaf or the plant. So it is osmosis which is facilitating the feeding in these insectivorous plants like the Venus fly. Explain briefly the role of osmosis in living tissues in animals. Explain briefly the role of osmosis in living tissues of animals. Now, osmosis in the kidney, it enables the reabsorption of water from the kidney tubules back to the bloodstream, facilitating the process of osmoregulation. Let's look at our diagram. In the diagram, we can see the arteriole from the renal artery coming into the glomerulus. In the glomerulus, there is a lot of pressure which forces the content in the blood capillary to enter into the renal capsule. That one is what we call ultrafiltration. So there is filtration occurring in the renal capsule between the glomerulus and the renal capsule. Now, when the content has been filtered or our filtrate, will move along the convoluted tubuli. Now, in the convoluted tubuli, still we can see the arterial capillaries surrounding the proximal convoluted tubuli. And now in the proximal convoluted tubuli, glucose and water are going to be reabsorbed from the kidney tubules back into the bloodstream. And that one is going to be by a process of osmosis. And as you go down to the descending limb of Henle, salts and more water are absorbed from the tubule back into the blood capillary. So it is osmosis which is enabling the reabsorption of water from the kidney tubules back to the bloodstream, facilitating the process of osmoregulation. In amoebas and paramecia, osmosis enables these organisms that live in freshwater bodies to absorb water. Water will enter into the amoeba and the paramecia by osmosis. So it is osmosis which enables these organisms in freshwater bodies to absorb water. Osmosis is also used or applied in food preservation. Osmosis is also applied in food preservation. One of the ways of preserving fresh food like fish is by putting it in salt. And when you put fish in salt, that means that the salt outside provide a hypertonic solution compared to the solution in the cells of the food or fish. That one is going to cause water to move out of the cells of fish into the salt solution. And the removal of water from fish is going to preserve the fish, because when you remove water, you prevent the fish from going bad easily. So osmosis is being applied in the food preservation. Explain what happens when plant cells are put in hypertonic solution. Explain what happens when plant cells are put in hypertonic solutions. The concentration of the plant cell sap is hypertonic to the solution water in the medium. The concentration in the plant cell sap or vacuum is hypertonic to the solution or water medium surrounding the cell. So what is going to happen is the cell will draw water inside by osmosis. That is number 2. Through the cell wall and cell membrane into the cell cytoplasm, which is number 1. The cell will draw water by osmosis, shown by arrow 2, through the cell wall, cell membrane, into the cell cytoplasm, which is number 1. The water will enter into the vacuum, number 1, by osmosis. It enlarges and exerts an outward pressure, which is number 3, onto the cell wall. And this one we call it the tag pressure. So when water enters into the cell vacuum by osmosis, the cell vacuum enlarges. And when it enlarges, it exerts an outward pressure, which is pressure shown by arrow 3, onto the cell wall. And that pressure is what we call the tag pressure. Increase the tag pressure, which is shown by arrow 3, pushes the cytoplasm against the cell wall until the cell wall cannot stretch any further. That pressure, the tag pressure caused by the increase in volume of the vacuum, it pushes the cytoplasm against the cell wall until the cell wall cannot stretch any further. The cell becomes firm or rigid and is said to be tagged. The cell becomes firm or rigid and is said to be tagged. That is what happens when a plant cell is put in a hypertonic solution. It will take in water, the vacuum will expand, exert tag pressure onto the walls of the cell wall, and the cell will become firm and rigid, and we say that the cell is tagged. As the cell wall is being stretched outwards, it also develops a resistant inwards pressure that is equal and opposite to the tag pressure, and this is called the wall pressure. So we are seeing the cell wall being stretched outwards, and when it is stretched outwards, it also develops a resistant inward pressure, resisting the outward stretch, which is equal and opposite to the tag pressure. And this pressure or resistant inward pressure created by the cell wall is what we call the wall pressure. Explain what happens when a plant cell is put in hypertonic solution. Explain what happens when a plant cell is put in hypertonic solution. The plant cell sap is hypertonic to the solution medium. The plant cell sap is hypotonic to the solution medium. Water molecules, that is shown by arrow 2, are drawn out of the plant cells by osmosis into the hypertonic solution, number 1, through the semi-permeable membrane of the plant cell. So we have placed our cell in a hypertonic solution, and the water now is going to move out of the cell of the plant by osmosis to enter into the surrounding solution, which is surrounding the cell, and that solution is hypertonic, and that one is solution 1. Through the cell membrane, which is a semi-permeable membrane of the plant. As a result, the plant cell will start to shrink, that is becoming less rigid, and will become flabby. The plant cell starts to shrink, going inside, as you're seeing in number 3, and it becomes less rigid and it becomes flabby. The cell membrane, or sometimes what we call the plasma membrane, number 3, is pulled away from the cell wall, and the cell is said to be flaccid. The cell membrane, our plasma membrane, which is number 3, is pulled away from the cell wall, and the cell is said to be flaccid. This process by which a plant cell loses water, shrinks, and becomes flaccid is called plasmolysis. The process by which a plant cell loses water, shrink, and become flaccid is called plasmolysis. However, the shape of the plant cell is maintained by the tough, rigid cellulose cell wall, which prevents crenation in the plant cell. The cell wall will not shrink. It will maintain the plant cell shape, preventing crenation in the plant cell, because the cellulose cell wall is rigid. Therefore, it maintains the shape of the plant cell. Explain what happens when animal cells are put in hypertonic solution. Explain what happens when animal cells are put in hypertonic solution. The concentration of water in the cytoplasm of the animal cell is hypertonic to the solution medium. The concentration of water in the cytoplasm of the animal cell is hypertonic to the solution surrounding the cell. Water molecules are then drawn into the animal cells. The water molecule will be drawn into the animal cell from the surrounding medium, and that one is going to be by osmosis through the semi-permeable membrane. Because the cell of the animal is hypertonic, water will be drawn from the surrounding of the cell into the animal cell through the semi-permeable membrane. The cell swells as water is drawn into them by osmosis. The cells will continue to swell as water is drawn into them by osmosis. As water continues to enter into the cell, the weak animal cell membrane bursts, the process called lysis. Because water is continuing to enter into the cell, the weak animal cell membrane is going to burst, a process we call lysis. As water continues to enter the cell, the weak animal cell membrane bursts, a process we are calling lysis. In the red blood cell, this process is called hemolysis. If we are using the red blood cell, this process is called hemolysis. However, in unicellular organisms like amoeba and paramecia, bursting of their cells does not take place because they have specialized organelles called contractile virtues for removal of excess water out of their body and cells. So what happens if you look at the diagram? When water enters into the cell of an amoeba, it will not swell and burst because it has got a contractile virtue. The water will enter into the contractile virtue, and the contractile virtue will move towards the cell membrane. And when it reaches the cell membrane, it will discharge the water outside the cell. And this one continues and continues, and the process continues. So you find that these unicellular organisms like amoeba and paramecia, bursting of their cell does not take place because they have got specialized organelles called contractile virtue, which removes excess water out of their body cells. Explain what happens when animal cells are put in hypertonic solution. Explain what happens when animal cells are put in hypertonic solution. Now, the concentration of the animal cell cytoplasm is hypotonic to the solution surrounding the cell. The concentration inside the animal cell cytoplasm is hypotonic to the solution surrounding the cell. Therefore, the surrounding hypertonic solution will draw water out of the animal cells by osmosis through a semi-permeable membrane. The water in the animal cell is going to come out, and it goes into the surrounding hypertonic solution through the semi-permeable membrane by osmosis. Continued loss of water causes the cells to become smaller in size, and their membranes become wrinkled. When the cells continue to lose water, they are going to become smaller, and their membranes are going to become wrinkled because they are losing water to the neighboring solution, which is hypertonic, through the semi-permeable membrane by osmosis. This process will continue until the concentration in the cell sap and the surrounding medium is equal, or it becomes isotonic. So water will only stop moving from the animal cell to the surrounding medium when the concentration of both becomes equal, what we are referring to as isotonic. The process by which animal cells lose water and shrink is called crenation. This process whereby animal cells lose water and shrink is referred to as crenation. Hello, my friends. Thank you very much for participating in Remedial Lesson 8, where we have talked about osmosis. Let's meet again in Remedial Lesson 9, where we shall talk about active transport. Thank you. Please please don't forget to subscribe.